Once our electrofocalization is done, we are now able to recover our cassette and our strips. So again, we apply the current with high voltage to allow the proteins to be separated. So now we take off the cassette along with the electrode body. And now we can recover our strips. What's covering the cassette is a film cover, so we need to remove that to be able to access our strips. We can hold the cassette firmly and try to pull and peel off from one edge to the other. So now our strips will be accessible. So now that the electrofocalization is done, we have access to our strip, and what we need to do is equilibrate it, because now the gel is in a buffer that's different than the buffer we're going to use for the second dimension. So what we need to do is solubilize the focus proteins and prepare them for the second dimension. So the way we do it is we pick up the strip from the negative end, and again the gel is on the top, and so we pick it up from the plastic side, and we need to insert it in a 15 ml falcon tube with the gel side always pointing up, so that way only the plastic side is touching the falcon tube. What we need to do now is to equilibrate it with the equilibration buffer number 1, which contains DTT, SDS, and urea. This buffer will reduce the sulfohydryl groups. So we add about 3 ml of this buffer, and we have to incubate with gentle agitation for about 10 minutes. So what we need to do is to actually put it in a Tupperware, and we can put it on a shaker that will allow for constant agitation. So after the 10 minutes are done, we need to discard the equilibration buffer number 1 and put the equilibration buffer number 2 instead of it. Now, equilibration buffer number 2 has iodoacetamide, and this buffer will alkylate the sulfohydryl groups. And again, we need to put it on a shaker with constant agitation for another 10 minutes. So for the second dimension, we have a special kind of gel. It's a normal SDS page gel that we use for electrophoresis, but this gel has only one single well, which will fit our strip, and it will allow us to place it here and let the proteins disperse into the gel as we apply a current. What's important is to leave a space for the molecular weight ladder, which will allow us to follow how the proteins separate. And what we need to do is to take out the green cone and cut it so that we have a little cone which would fit the size of the molecular weight ladder that we're going to load on the gel. So we can put our strip in, but we'll have some spaces between the strip and the gel, and the proteins that will exit the strip will disperse into the buffer instead of going into the second gel. So we need to seal it by putting some molten agarose to seal these spaces. So here we have on the hot plate some boiling water, and we have prepared low melting agarose in SDS buffer, as well as we have added trace amounts of bromophenol blue, which is a dye that will allow us to track the ion front during the run. So we make sure that the agarose is well melted, and we put it out just a few minutes before we start to make sure that the agarose is not very hot. As was mentioned in the previous video, we need to make sure we have a little space for the molecular weight ladder. So what we need to do is take off the green comb and cut one little piece from one extremity, about 4 to 5 millimeters, that will fit our molecular weight ladder and allow us to visualize where the protein separation. So then we insert the green section back into our well. The second point we need to be careful of is we need to dry off our excess buffer that's still on the plate after we take it out of its package. So we can get a little tissue paper and try to dry off the excess buffer, especially inside the well, because when we insert our strip, we need to add agarose in order to seal the spaces. So we dry it off, and once we make sure that there's no buffer inside the wells, or we have dried it off enough, we can now insert our strip. So what we need to do is to take off our strip from the equilibration buffer that was in, and place it on top of that plate, just to make it easier for us to place it once the agarose is in, so that the agarose doesn't solidify before we put our strip in. So now that our equilibration is done, we can embed our IPG strip into the STS gel that we have here, and we can add agarose and able to seal the spaces between the two gels and allow the proteins to separate. So we can pick up our strip from the negative end and make sure not to touch the gel end, so just touch it on the plastic end. And now we can cut the plastic end from the positive side so that our IPG strip can fit, because remember that you have added a comb for the molecular weight marker. So in order for the IPG strip to fit right into the well, we need to cut that little section, and then we place it on the longer curved end of the STS page plate. That way it's very close to the well, so that when we add the agarose, we can just press it into it to embed it. Now we have to make sure that the agarose has cooled down fairly well before we can put it in. And then we can take about 400 to 600 microliters of agarose, and we can load it into the well. It's okay if we get any bubbles, because we can push them out by when we actually insert the IPG strip in. The way to insert the strip is to push from one side and try to take out the air bubbles, and then keep pushing along the IPG strip till it goes right in, and we push it all the way to the bottom, making sure there's no air bubbles or anything that's in between the two gels, and that way our proteins will be able to separate easily when we apply a current in the second dimension. We also make sure there's no agarose going into the green comb area here, so that our marker can be loaded onto the gel too. The blue color will enable us to follow the migration of the proteins on the gel. So now we can wait till the agarose solidifies and we're able to run our second dimension. So now that our agarose has solidified, we are able to load our second dimension to separate proteins according to their molecular mass. So this is the electrophoresis apparatus that we're going to use, and it has 2 electrodes as usual. So our gel here contains the solidified agarose with our strip that has the proteins focused according to their isoelectric point, and we have here the comb that if we take off we'll have a space to load our molecular weight ladder. So now we can load our gel onto the apparatus. So we're going to first explain a little bit how it works. So it has two electrodes. The first one is the negative electrode, which is connected to the bottom over here. So this is a platinum wire. It's the first electrode. The second electrode is the positive electrode, and the platinum wire is at the bottom under the green rubber. So we'll explain later how this electrophoresis works, but now we are going to load our gel with the longer plate pointing to the exterior. This unit usually fits 2 gels. If we don't have another gel, then we can put the plastic plate and close the unit. As we mentioned before, the negative electrode is right at the bottom in the middle here, so we can fill out that compartment with our buffer, and what will happen is that the current will pass through the negative electrode through the wire, and then the electrons will leave traversing the buffer and then get into the gel or RPG strip, and will separate the proteins, and it will go all the way to the positive wire and go back to the positive electrode. So the buffer will provide the electrolytes to connect both electrodes. So now we are ready to load our second dimension, and we can put our cassette into the cell and add our STS page running buffer in the middle chamber, as well as the exterior compartment, just to cover up the bottom electrode. Once this is done, we can now add our marker, which goes into the well we created earlier. So our marker is a molecular weight ladder for our proteins. It contains a loading buffer, which will allow it to sink to the bottom, and a dye that will allow us to follow the migration. Once this is done, we can add the lid and close our electrophoresis unit, and then we can attach our 2 electrodes, red to red and black to black, into the power pack. And once we apply a voltage, we can see bubbles that will indicate that our electrophoresis has started, because we have the electrolysis of water. So after a longer period of time, we will see that the marker has moved along with the proteins. So this migration will conclude the second dimension of our gel electrophoresis. So the next step is to color our proteins in order to visualize them.